Hello. So I've been working in metabolomics for 15, 16 years. My background's in machine learning and statistics. I'm actually speaking 3 times at this conference, so this is the first of 3, which is mainly on study design. Tomorrow I'm speaking at the biomarker workshop, where I'll be talking a bit more about untargeted metabolomics. And then I'm speaking, Thermo have paid for me to come to this conference, and I will be speaking for them on Wednesday lunchtime, more about quality control and reproducibility. So this just gives you an idea of over the last 20 years, I sat down and had a bit of a brainstorm about how much time I really spend at each part of the workflow that you would associate with a statistician. And it turns out the majority of the time is spent doing study design, helping people design their study, how to collect samples, how to set up a biobank, how to finalize SOPs, and then a significant amount of time designing analytical experiments involving pooled QC samples, making sure you randomize samples, making sure you're not going to succumb to batch effects, etc. And then, as anybody who's done untargeted metabolomics knows, there's a big part which is to do with data cleaning and deconvolution. And when it actually boils down to it, I only spend about 15% of my time doing the statistics. So a statistician doesn't really do that much statistics, and you should really bear that in mind. And if you want to do a study, really buddying up with the statistician right at the start is a really good idea, because they will help you make your results a lot better by letting them help you design their study. So, as Tim said, I've been asked to talk about study design, so I'm going to leave the more things I usually talk about to some of the other speakers, particularly Sasty's going to talk about quality. But study design and biostatistics have a huge interplay. Unless you know what your study design is, you don't really know what biostatistics are going to do, and if you don't know what your biostatistics capability, that can really constrain what you do in your study design. So it's a bit of a chicken and egg situation, and it's sometimes quite difficult, and this is why it's really, really important to do pilot studies before you really get into the meat and potatoes of a project. But the one thing that does link them quite strongly is the idea of uncertainty, and from uncertainty, the assumptions that you make during your statistics and during your study. And really, statistics is the study of uncertainty, whereas the design of experiments is the reduction, or to try and reduce that uncertainty when you're collecting the data or designing your protocols. So one really does is they really are symbiotic in that respect. Uncertainty really need to break down into 2 different sorts, bias and variance. Where bias is the systematic difference between the measured value and the true value, the real absolute value. And this is usually estimated by the mean of repeated experiments. So you repeat experiments, you'll have a standard, and then you can calculate how far away your repeated value is from that standard value. And that will give you an idea of a bias. Variance is more about the noise, so the random variability measured on a value for a given entity. And this is usually estimated by a measure of dispersion, for example, the standard sample standard deviation. And just to visualize that, this target, you will see, if you Google bias versus variance or experimental design, you'll see this plot comes up all the time. So this is nothing novel from me. But it's a very good example of how you can see the ideal situation where ideally we would like to be here. We would like to have a really, really low dispersion when we do repeat measurements, but we would like to be really, really accurate. So what we measure, we would like to be true. What we really don't want to be is down here. We don't want to have high variance and be completely inaccurate. Bias depends on what you're doing. If you're doing a pilot study and the bias is something that you understand and is reproducible bias, then that doesn't necessarily destroy a project. And variance this is something which the majority of this lecture is going to be about, and the idea of how to reduce or manage or understand the variance in your study design. Just to quickly talk about repeatability, and this really does flag up the idea of how bias can be really negative. If you repeat a study, this happens a lot in metabolomics. You'll be surprised the amount of data sets that I've been given once I've dug into them and asked around. And one example, NMR data set. The controls were collected from a single hospital. The diseases were collected from a different hospital. The controls were run on the NMR in March. The controls were run on March. The disease were run in July. The controls were deconvolved separately from the diseases, and then they were all put together. And guess what? We had a really good biomarker. ROC area of 1, no confidence interval required. Fantastic. But unfortunately, there's a 100% confounder in 3 points in that experiment. Metabolomics, untargeted metabolomics and semi-targeted metabolomics isn't that reproducible. You cannot just run samples whenever you like. You have to carefully design your study and make sure that you run all your samples in a series of randomized batches. And I believe one of the other speakers is going to delve into that a bit deeper than I. But I just wanted to point out, in terms of the bias and variance, this is how you could represent that. But obviously, you can still have bias. If your bias is really reproducible, i.e., you just have an instrument that measures things slightly off, or your methodology measures something slightly off, but it does that repeatedly, then that really, in a case control study, is not an issue. Or if you're repeating a case control study and you're doing a validation test, it's not disastrous. You want to avoid it, but it's not disastrous. Now, generally, the bias variance, there's a trade-off. And I've pulled this slide from my stats lecture. So this is very prominent when you're talking about optimizing, say, a PLS model where you... I'm not going to do a stats lecture, but you understand the concept of doing cross-validation and picking a number of latent variables for a discriminant analysis. And the idea is that you're trying to get a trade-off between doing a good fit but without overfitting. And that is pretty much the trade-off between having bias and variance. And this interplay exists at every stage throughout the experimental process. And it's always a balance between not overly focusing on individuals, but also not introducing too much noise. And just to flash up a bit of variation in terminology, this comes from this paper down the bottom, because there's a lot of terms for the same thing. I'm kind of talking about bias, uncertainty, and standard deviations, but analytical chemists might talk about trueness, accuracy, and precision, and actually the type of errors would be systematic and random, and then the sum of those two. So this is kind of the math. This is how I'm going to talk about it, but this is what a lot of people in the room probably would understand in terms of their experiments. But you can see that we are talking about the same thing. Now, if you don't do anything after this lecture, anything more than one thing, read this paper. It's not particularly about metabolomics, but it's a really good description of how bias and variance is introduced all the way through an experiment, from sampling, which is what I'm going to talk about today, measurement, I'm going to touch on, modeling, which I'm not going to talk about at all, but there's other workshops on the modeling. Even when you get to doing your hypothesis testing and the way you report your data, you can introduce bias and variance at every point throughout that, either accidentally or intentionally or unintentionally. That it exists, and it's all about the idea of experimental design, and in a way statistics is about either understanding, controlling, and reducing all those things. So for the last 10 years, I'd say most of my research has been clinical, so there's always a bit of a clinical slant when I talk about sampling bias, but hopefully anybody who's a plant biologist or environmental biologist could kind of extrapolate from the examples that I give. Specificity bias is sometimes a really, really good thing. When you've got small sample sizes, an example, before I was at Edith Cowan, I was at the University of Alberta, which has the highest prevalence for multiple sclerosis in the world. And Alberta has the highest prevalence of multiple sclerosis. So we did some studies where we were looking for biomarkers for MS, and we just took samples from people in Alberta, which is a really nice controlled population. They all have a fairly standardized lifestyle, genetically similar-ish. So it's quite a specific population, and we know that they are particularly prone for it. So we could come up with a fairly good biomarker. But the problem with that is you could come up with a good biomarker, but it is only a good biomarker for people with MS in Alberta. You can't generalize to the rest of the world. So specificity can reduce variance and increase effect size, but it's difficult to then generalize from it, or you'll have to do a lot more work to make sure that it's generalizable. Obviously, the non-representative control population is massively important. The only thing that should be changing when you're selecting your cases and controls, the only thing, the difference between the individuals should be that disease or that sign or that symptom. Matched by age, matched by gender, matched by BMI. These are all the important things in metabolomics. Not matching for BMI is one thing that like the newbies kind of miss, but anybody who's done untargeted, certainly LCMS, finding biomarkers for obesity is very, very simple. And if that is 100% confounder or a very big confounder in your disease study, then you really will be finding biomarkers for something that's not the thing that you're interested in. Often we'll try and, certainly for very rare diseases or very particular diseases, we'll even ask siblings to be the control, so that there's some genetic similarity. But again, then you might have difficulty with age matching and etc. So again, you just really have to sit down with statistician, epidemiologists, the people who are running biobanks. So if you've got access to a big biobank and you're doing a nested study where you can select people from a bigger population, then that's fairly straightforward. So sample timing, again, I gave the example, don't run your samples 6 months apart. You will find that biomarker for running things 6 months apart. Location, ideally, collect prospectively, design a study, and run it retrospectively. So I did a big project looking for biomarkers for preeclampsia, which has a prevalence of around 3 to 5%. So we had a prospective study which ultimately collected samples from 5,000 women at 3 time points. So the only way we could do a proper study in metabolomics is we had to collect all the samples and then go back and find enough cases that we could then match to do a biomarker study. And you can imagine with a prevalence of only 3%, we didn't actually have a very powerful model even with 5,000 women. So that can be quite difficult. The rest of the things are pretty general. I want to skip on or else I'll be talking forever. Measurement bias. I think the other guys are going to talk a little bit more about this, but the obvious things are consistent sample handling. Don't collect the cases in one type of tube and the controls in a different type of tube. We can find biomarkers for the plastics. We can, again, time to collection, if really consistency is the thing. The time you sample prep, the time the samples are left on the bench before they go through the instrument. All these things can introduce bias and variance as well. But bias will really kill your project. So now I've got you here, I can get you to do some maths. So I like to, a few people hopefully will have seen this before. I presented it in Ireland. Well, parts of it anyway. So I really want to talk about sampling variance, because this is really important. The rest of it is unbelievably important too, but quite obvious. Finding bias, once you think about it, is fairly obvious. Understanding the problems with sample variance is not quite so obvious, even though we all get taught it at high school, and it's the basis of statistics. So let's just hypothetically suggest that there's 10,000 people with a certain disease. Let's call it Broadhurst disease. So there's 10,000 Broadhursts in the world, and they've all got a genetic disorder. Now, when you're doing a study, you would be unlikely that you would be able to collect samples from all 10,000 Broadhursts around the world. And let's say we think we're pretty good, we're going to do a sample size of 50. So what you do is you select 50 from that, and then normally when we're doing the statistics, we're comparing means. In this case, we just want to see whether the mean is different from 0. So we've done a sample of n, collected n patients from that 10,000. We've calculated the mean, and that's the green spot. And the green spot says the mean in, say, metabolite X is 4.1, and the mean in metabolite Y is 4.2. Now, does that really tell us very much? Would you trust that that is the true value for the whole of the population? Would you trust that X is 4.1 and Y is being 4.2 is being correct? Well, obviously not, because I've written it at the bottom. So it's a bit of a giveaway. So whenever you're doing a subsample, you're not going to get the exact answer, because you're sampling, and even if you're randomly sampling, you're sampling from that whole population, and it will never be completely the true answer. And this, again, is why we do the statistics, to see how we can assess how uncertain that measure is. So what I'm going to do is I'm going to show, if you do that experiment of 50 100 times, what happens. And there we go. And you can see each experiment. So we're going out, we're finding 50 Broadhursts, we're collecting the measurement, and we're calculating the mean, and we're doing that over and over and over and over again. And you can see that the value, it's kind of somewhere in a pretty safe region. There is a big splodge, but there's a certain amount of variance. And you can see, it's looking like a pretty systematic shape. And then that's the end point. And if we look at the numbers, which you can just see, they're not exactly the best resolution, but you can see that the mean of the whole population was 3 and 4, and then the means of the means, very important, it's the mean of those 100 means is exactly the same as the mean of the whole population. You do multiple experiments and average them, you will get the same mean as the true population, whatever study you look at. More importantly, if you look at the standard deviation of those, and then you divide the standard deviation of the true population by the square root of the number of samples that we take each time, which is 50, that is exactly the same as the standard deviation that I've just done with this computational experiment. So you repeat an experiment, you can get an idea of the true distribution of the study. Now, what happens if we change the sample size? So this time we're going to do 10. And you'll see that we're getting the same kind of idea, but this time the average value is really flying around. Sometimes it's nearly up to 5, to 10, to 8, and sometimes it's getting close to being a negative value in X, and I think there is a negative value eventually. So any single experiment, you'd be really tough to decide whether the mean value for this Broadhurst disease in X is above 0. And again, we can go through the maths, and the standard deviation sticks to the formula. Standard deviation of the original population divided by square root of n is equal to the sample standard deviation of all those mean values. If we do this for different numbers of samples, starting with 10 all the way up to 10,000, you can see that eventually it gets smaller and smaller. So that confidence interval goes smaller and smaller the bigger the sample size. And this is the central limit theorem, and this is the basis of all the statistics that you do when you're doing t-tests, etc. And then what we generally do, we're all trying our hardest to move people away from p-values and towards confidence intervals. And what you would do is calculate a 95% confidence interval, which is the mean times 1.96 times the standard error. And if all the assumptions you've made about the data normality, the standard error is equal to the population error. And that's pretty much what you do when you're doing a t-test. And if that confidence interval is bigger than 0, is above 0, then it's significant. If they're both below 0, then it's significant. If 0 sits somewhere between that confidence interval, then it's not a significant finding. To say, you can extend this to actually get a p-value if you want, but far more fun. So, if we did this in terms of a biomarker, and again we've got an N of 50, and we've got an effect size of 10, and you can see I've shaded out where that 95% confidence interval of each of those means, and they're pretty separate apart. You would say these two things are significant. Now, if we only measure 3 samples, same effect size, same distribution, you've just sampled it less, that's the confidence interval. So this is where you have to understand that the biology, the sample size that you collect has a massive effect on whether you see that the biology you're measuring is real, there's a real effect or not. Just because something isn't significant doesn't necessarily mean the actual real effect isn't important. It's just for that sample, and for that variance, and for that effect size, it might not be. So, p-values are rubbish. This is why. So here's 3 studies, same p-value. The top one is a model system. Massive effect, huge effect. Say it's, I don't know, an animal, a mouse model, and you've prodded it with a electrode or something. You're really scared the living bejeebies out of this animal. They've all been kept and fed in very standardized environments. So the variance, the biological variance is very tight, and then you really hurt these animals, and that's why we do animal models, unfortunately, and you get a big effect size. So you can get away with an N of 3. for this study to get a really significant value for the same p-value. Now, and the flip side, if you're doing an epidemiological study where there's a massive amount of heterogeneity about the populations you're looking, the effect size might be really small. So in order to get those two confidence intervals apart, you need 3,000 values. Where we kind of sit in metabolomics, certainly in clinical, is somewhere in the middle, where you have a reasonable effect size. There's a certain amount of variance that that's unavoidable, and we're dealing with the ends of 30 to 100, generally in a kind of pilot study area. So you can see that that same p-value, you could have completely different parameters for your experimental design, and it will give you completely different results. Now, I always get asked to talk about power. I try and avoid it because it's always confusing to talk about, and people want to know how to, what's the sample size, how to power. So I've pulled this from this fantastic paper. Again, I'm going to have a go at just pitching this in 2 seconds, but read this paper, it's really good. It's about how small sample size are a bad idea, which is something I'm very passionate about. The slides are going to be given to EMN, I think, or they're going to be available online, so you don't have to write everything down. So the concept is, when you've got a null hypothesis, you've got an observed distribution. For it to be significant of a p-value of 0.05, I've just explained to you that that's equivalent to that 1.96 times the standard error. So if you've got a p-value of 0.5, it's exactly 1.96 times the standard error away, and that is where the distribution is, if you're assuming that's a representative sample. Now, if you then assume that that's correct, that you've got enough samples in that study design, that effect size, that distribution is very, very accurate, and that p-value is static, and then you repeated the study based on the same sample numbers, then you would get, again, this is what this is kind of figuratively displaying, and this is showing you the number of, so p-values you're generally worrying about false positives, so in this sense, the 5% false positive will be this bit here, and in here we're talking about false negatives. So power is all about when you find something and that's real, but you assume that it's not real. And unfortunately, if you do this, if you follow the regime that I've just described, where you have the same sample numbers as the experiment before and you've just got a p-value of exactly 0.05, then you've got a 50% chance when you repeat that study of dismissing something that's actually true, because it will be behind this line, and if the p-value of 0.05 is that line, if you have a mean which is just to the side of that line, it will say it's not significant. But we know from that this is the distribution of the true values, that if it's behind there, it really is true, but the statistics will tell you that it's not. So you have to be very, very careful, and yet one of the ways to make sure that you increase the power, so you reduce that probability of a false negative, is to increase the sample, is either increase the sample size or reduce the variance. And that's what this paper is talking about. So I recommend that you go and read it, because it's very difficult to talk about in just a couple of minutes. But the take-home message is the power and statistics, it's affected by 3 things that interact: the effect size, the sample standard deviation, and the number of samples. Now, again, metabolomics is very, very, very difficult to estimate how many samples. If you read the paper that I wrote with David Wishart's group, we do talk about ways of calculating it based on, certainly for biomarkers, based on what your expected outcome would be. But if you're just trying to look for, if you're doing a pilot study and you just want to see whether you've got a number that would be reasonable, an N of 35 is considered a reasonable starting point based on this curve, which is the reciprocal of the square root of n, which is related to that standard error. And you can see that when you get above 35, obviously more than 35 is great, but what you gain from having more, this is for a single biomarker, remember, what you gain by having more samples is reduced, whereas if you're really back here, suddenly the power disappears. So if you're only looking at 5 samples, you really are struggling, or less. Again, this is just a very arm-wavy number. If you're going to do cross-validation in your modeling, then you really want to add on a third for that to take into account that you're going to be doing some sort of fold validation. Really, this is going to be covered in my talk tomorrow. When you're looking at multivariate statistics, this number goes out of the window, and you really want to be going up to the thousands, and that's why we're doing the phenome census. So, kind of one last concept is the idea of marginal gains. And this, again, it all ties in with this idea of power, the variance, the effect size, the sample size. If you have a fixed effect size, that's generally in a study where you're just observing people and you're not really or not inflicting some sort of treatment on them, like exercise or diet, but you're just observing people. Often you just have a fixed effect size. And sometimes, due to the prevalence of the disease, you might only have access to 20 samples in the time frame of your project. So the only alternative you have then is to reduce your variance so that you can make something that's maybe not significant. Now, you can do that during the study design, like I talked about. You can focus on a subset of the population, so you could just look at all the Broadhursts that are over 65 and obese and talk too much, and that would be a lot tighter group than if you looked at the whole, and you would hope that the variance would be less, so you might see something significant. And remember, in metabolomics, we're very much doing hypothesis generation, so finding something and then relaxing out, it's hard to find stuff. Then you can measure it really accurately, reducing the technical variances, and then you can relax it out and maybe look at a bigger population. So when we're doing untargeted, very noisy analytical methods, we really want to focus in on the crux of the problem, because then when you go to a targeted method where you will have far more reproducible data, then these things can be relaxed a bit, because hopefully your biomarker will be a bit more clear. But hopefully the picture kind of describes what I'm trying to do here. You reduce the standard deviation, you can see you go from being not significant down to significant. So understanding and reducing the sources of variance in the study is of equal, if not more importance, as the number of measured samples. So they're both important, but really trying to reduce the variance in a study is very, very important. So if we just look at the standard, very basic overview of workflow, we go from the idea, the study design, talk about what patients you want to be looking at, or what plants you want to be looking at, and what locations they are, how you recruit them, or how you collect them in the field, how you collect samples, or how you collect, water samples if you were looking in rivers. Biobanking, very, very important. We can't, unless you're doing a targeted method, you can't run samples on the fly. You have to collect, biobank, do a very structured experimental design, and then run them through in batches, and then make sure that all the variances are moving. Quality assurance, I did a lot of work on using pooled QC samples to assess and correct batch drift. I'm not going to talk about that today, but I will be talking about that on Wednesday. And then obviously the data pre-processing and stats, and then hopefully a publication. But if we start using statistics and using our brains and really thinking about it, you can reduce just a fraction each of those areas. You will be surprised how much you will gain by just being very rigorous at every stage. A lot of the things that I describe might seem like I'm nitpicking, so I'm just... Okay. But they really, really are important. And just taking a small amount of error out of a study, and really, over and over, this is stuff that myself and Rick Dunn have been doing for 10 years, is we keep redoing our workflows, we keep redoing them until we get them as efficient as possible, because we know that that has a massive effect on how many samples we have to run and how much a study is going to cost. So it's really, really important. And the byproduct, every point of that workflow, you collect data, and that data can then help you guide you in your statistical analysis and certainly in the publication. Which gets you to this kind of more of a biostatistics workflow, where we're collecting data at every point, and we're doing tests and bias checks, and checks for normality, and checks for quality, and then ultimately getting down doing some stats. And then the stats might feed back in with some logistic regression that might be correcting for some of the bias that you've discovered post hoc or is unavoidable. So there are ways mathematically that you can use that information later in the game. So just to conclude, Marginal gains, really important. Repeatability and reproducibility, important, but it depends on the study. Try and remove bias, try and remove variance. Standard operating procedures are the standard. Quality control is really, really important. Better data, not necessarily more, but large data sets really are helpful, particularly if you're going to start doing multivariate statistics with any meaning. And I think I have to acknowledge the fact that my lab over at ECU is a Thermo Fisher proof of concept lab, so partially sponsored, and they have sponsored me to come to this conference, so I acknowledge their sponsorship. So I haven't really biased anything in this talk, but maybe later on. And yeah, come to the sunniest city in the world. You think Brisbane's nice? You really need to come to Perth. Feel free. So on that, I think I'll finish. I think just about hit on time. Okay.
Okay, good afternoon. Hello, everyone. Thank you so much for coming here on a Sunday. I'm really excited to start this year's meeting with so many people from the EMN. So as has been introduced, I'm Sastia. I'm the first chair of the EMN, so I'm really, really happy that after the EMN was first established several years ago, now we have a really active EMN, and also it's expanding. So I'm really feeling a really good energy here, and hopefully this good energy continues throughout the conference here in Brisbane. So we have already finished David's talk on the experimental design. So today I would like to talk more on the practical sampling and the approach and how to choose appropriate analytical platform. So as we all know, metabolomics is a multidisciplinary science. Probably many of you here come from a background of analytical chemistry, biochemistry, and some come from informatics or statistics. And I myself, I have a background in biology. So when I first started working on metabolomics, I had a lot of difficulties at first to understand, for example, the statistical concept and also the informatics side of it. So as we have seen a lot of this kind of omics scheme, I think a lot of field of omics probably are less complicated in terms of the variability of the samples that we analyze. So the metabolome is really complex, and we analyze so many different kinds of metabolites. It contains, for example, sugars and also lipids. They all have very different physicochemical properties. So how to choose appropriate approach and appropriate analytical platform is really, really crucial to start a successful metabolomic study. So a typical workflow of metabolomics analysis involves, first of all, we have to do sampling of a biological sample. So your biological sample can be anything from microbe to higher organisms, plant, and also human subjects. And afterwards, we go for sample preparation. Depending on the type of biological samples that you're working on, then you will have to do different types of sample preparation. Yeah. And for example, you have originally liquid samples, or even part of plants, different part of plants have to be treated differently. And also, we go to the sample analysis. This is where the data acquisition starts. And then once we have the data, then we have a lot of very complex data. Then how to translate that complex data into something that is tangible and we can easily measure and understand. So usually we create this data matrix, and then afterwards subject that to different type of multivariate analysis to visualize our data. So the experimental scheme of metabolomics involves several steps. Today we are going to talk about experimental design. And in the experimental design, I think we have different points that we have to consider to start a successful metabolomic study. So afterwards, we have to also consider, after we have conceived the design, then we have to consider appropriate sampling preparation method, sample preparation method, how to do data acquisition, how to do data pre-processing, data analysis. Afterwards, we go to biological interpretation. But before we start all this, because what we're analyzing is biological sample, we have to start by asking the right biological question. So sometimes I have a lot of collaborators from biology, basic biology, and they come to the lab and they said that we want you to analyze our samples. And when we ask them, what is your study purpose, sometimes it doesn't really match with what metabolomics can do. And without understanding the metabolomics power and what metabolomics can be used for, I think the use of metabolomics will be suboptimal. So by asking the right biological question, we have to understand the organism of choice, and we have to construct a very good working hypothesis. So first we have to start with a good working hypothesis, and we have to understand whether we want to go for the targeted or non-targeted approach, and then we then choose appropriate analytical instrument, appropriate sample preparation method, how to validate our data, and then data analysis method. Everything here, these points of considerations really affect the quality of the data, and also it will really affect whether the data that is generated from our study will be actually useful or not to answer the biological question. Without that, then usually it's like junk in, junk out. So you put a lot of effort, you do a lot of measurements, but then nothing in that data you can use. So for the analytical design in metabolomics, I think probably most of you are familiar with different approaches of metabolomics, but probably some of you are beginners. So we can have a targeted approach or a non-targeted approach. For targeted analysis, of course, the data quality is really good. We do a precise and quantitative measurement, but only a limited number of known metabolites. So first, if we want to go targeted, then we have to understand the type of questions that we want to ask in our biological system, and whether specific metabolites or specific class of metabolites can answer that biological question. If the specific class of metabolites can answer that biological question, then it's basically like we can validate we can answer the hypothesis using only targeted analysis. But then if we want to go for non-targeted, we don't know actually what we're looking for example, some type of studies like biomarker discovery, then we go untargeted, high throughput measurement of large number of metabolites. And also, if we don't aim to actually annotate the metabolites, we just want to know the whole metabolites that are scanned are treated as one fingerprint, and we just use that for discriminant analysis. For example, we just want to know what is the difference between one sample and the other. We can just treat that and go for metabolic fingerprinting. So the important consideration here is that we have to understand there's a very important trade-off between metabolite coverage and the quality of metabolite analysis. So here is taken by a paper by Thethoe in 2004 from Current Opinion in Plant Biology. Of course, if you do a single compound assay, you have a very good data quality. You have a very high quantification accuracy, sensitivity, as well as identity, you are sure. But then the higher the number of metabolites that you want to cover, then the quality of the data becomes lower, much lower, the resolution. But it depends on your study purpose. If you want to, for example, go for targeted analysis, as I mentioned, for example, if you want to do amino acid analysis, you know that what you want to answer is in the amino acid pathways, then maybe you can just target a single class, and then you focus on that class of compound. Then you can develop a very good and powerful analytical platform with high accuracy and high quantification accuracy to measure those metabolites. But then if you want to involve multiple classes of metabolites, as I already mentioned in the beginning, it will involve different different classes of metabolites, and they do not have similar physical-chemical properties. As we know in organic chemistry, then we have to either combine our analytical instruments, or we have to be really smart on the sample preparation and sample extraction method that we are going to employ in order to visualize or cover all of those metabolites coming from different classes. So for the sample preparation, it varies depending on the sample. I work with microbes. I'm a trained microbiologist, but recently I also work with plants and also animals. So I'm also learning because I can consider myself still a little bit in the early career. So first, my training is in microbial metabolomics, and for microbial metabolomics, I know a lot about microbes and its physical-chemical properties, so I can better design the type of cultivation protocol and how to do the extraction depending on different microbes. But then when I started to expand my research group, now I also do analysis of fruits like mangosteen, tropical fruit, banana. I also do coffee metabolomics, and I do shrimp metabolomics now. So as I go further and then expand, I realized that the complexity and the different type of biological samples have to be treated in a very unique way. And so the sample preparation normally involves like harvesting the biological sample, and then we do quenching. Quenching is a very important step in a metabolomic sample preparation. The purpose is to stop biological reaction in the cell. We have to immediately arrest the metabolism in the cell at a given time point, and it's very important to do that correctly. Otherwise, when you're doing, for example, different replicates, and you're doing it in a different way, you're comparing different things. As you know, that metabolomics metabolomics give a very sensitive, it's very sensitive to environmental perturbation. So if you treat your samples differently, then you might get different results, and resulting in very high standard deviation among your samples. Especially when you consider about microbes, the turnover rate of metabolites and the growing speed of microbes is really fast. So it's important that the quenching is done within seconds. So if your sampling preparation is not optimal to stop the metabolism really, really fast, then probably what you're getting is not a good quality of data. So in microbes, for example, like gram-negative bacteria and gram-positive bacteria, gram-negative bacteria is more prone to metabolite leakage. So in the case of E. coli, for example, if you want to employ cold methanol, which means you have a very cold methanol solution, and then you just inject the samples there, and then you take the sample pellet. That one is really good for microbial sample because it's really fast. It's basically just pipetting and then put it in the cold methanol or cold ethanol. But in that case, E. coli cells, for example, is very prone to metabolite leakage. Then you have a problem of metabolite leakage, and then the results of the intracellular metabolite pool that you wish to quantify will not be accurate anymore. So then we have also a method of fast filtration. So basically, stopping biological reaction in the cell can happen by separating the cell with the medium of growth. So in the case of plants, sometimes just rapid freezing with liquid nitrogen can do the trick. And for extraction, the purpose is to obtain metabolites in the cell. And depending on the target metabolite that you wish to cover, then you have to extract differently. Because you know the principle of organic chemistry, if you want to measure hydrophobic compounds and hydrophilic compounds, you have to extract them using different solvents. So for example, if you're aiming for hydrophobic compounds, for example, like hexane is probably a good solvent to use. But if you wish to obtain polar metabolites, for example, for GCMS, maybe methanol or water is a good extraction solvent to use. So for the data acquisition itself, analytical platform metabolomics research, usually there are two major groups in metabolomics, either the NMR metabolomics group or the MS-based metabolomics group. I personally work a lot with MS, but a lot of, probably some people here, can I have a raise of hand, who here works with NMR? Yes, and who here works with MS? Yeah, we can see that it's a majority. So I will explain the reason why. NMR-based analysis is really good because it's non-invasive. You can recover your sample, and analyte separation is not required. So it's really good if you want to analyze many kind of samples because there's no problem usually with if in the MS, if you analyze a lot of metabolites, and then sometimes there's a problem with carryover, and then many problems with reproducibility as well. But the problem with NMR-based analysis is low sensitivity, and the metabolite that you can cover is rather limited. But sample preparation is relatively easy. For mass spectrometry-based analysis, well, it's invasive. Usually sample is not recoverable in general, and analyte separation is required. And of course, we have to do the extraction and everything. Sometimes we have to do derivatization for GCMS. So sample preparation is relatively more tedious, but it has high sensitivity and selectivity. So the points to consider when selecting analytical method in metabolomics, we have to consider metabolite coverage, resolution, sensitivity, quantifiability, reproducibility, and high throughput. So when considering these points, when selecting analytical method, then mass spectrometry obviously has several good advantages and why it is mainly used in metabolomics. Well, it has high sensitivity, very good selectivity, and also it can connect with various chromatography, so the metabolite coverage is quite wide. But the weak point is sometimes we have ion suppression problem, and the quantitativity is not good. So usually in metabolomics right now, we use what we call the not the absolute quantification, but we measure relative quantification because due to the problem of ion suppression. And also reproducibility. When you measure your samples today and you measure your samples next week, as already is illustrated, sometimes it gives very different results. So when you analyze many samples in one batch, it's not good because sometimes the first sample that you measure and the last sample you measure may give different measurement, even though it's the same sample. So sensitivity sometimes decrease over time when you're analyzing in a long period of time. So it's really important for you to consider having a very good QC procedure. So quality control procedure, so you always have to have QC samples when doing a run for MS measurement. So using the QC sample, this somehow can correct the differences of variation within batch. And when we also consider the comparison of metabolome data that is taken in different batches, very careful study design, considering statistical consideration is very important as well, especially if you're running a lot of samples and then afterwards, next month, you're running a lot of samples again, and if you want to compare the data, very careful consideration has to be made in order to get very good quality of data. So when you decide the analytical instrument, as I mentioned, we have to consider like polar character of the metabolites and sometimes molecular weight. So depending on that, I put here a map. So different type of metabolites can be analyzed using different instruments. In my study, usually I would like to get as many metabolites as possible. So I do both GCMS and LCMS and then combine the data. But sometimes combining GCMS and LCMS data is also tricky. So I usually like to analyze GCMS data and LCMS data separately, and then probably they will give me different results. Or sometimes because there's an overlap of metabolites, if the metabolite, the trend is really strong, then sometimes we can still see that same metabolite affects the separation between samples. So to decide on a suitable analytical method according to the purpose of research and type of samples in metabolomics, this is just a graph. For example, we have whole cells or biofluids, and we have to do the fractionation, or we can just analyze the whole crude extract. And there are many different ways and many different analytical platforms that we can do in order to achieve the study purpose. But of course, we can see that we can do the sample preparation and we can select the method suitable for our study purpose. There are so many different options out there. So for the application of metabolomics, I would like to illustrate a case study for microbial metabolomics using a non-targeted approach for biofuel production. So the application of metabolomics for metabolic engineering is very, very few. So most metabolic engineers, they do not. In the beginning, when I started a research collaboration with a very famous metabolic engineer from the US, Professor James Liao, it is funded by Japan and US. It's a 5-year project, very big project, to use metabolomics for a low-carbon society. So the beginning of the use of metabolomics, we had a lot of difficulties because sometimes we give a lot of useful feedback that we think is useful, but for them it's not relevant. So the first couple of years to combine these metabolomics for metabolic engineering require a lot of intensive discussion, and also we have to carefully construct the study design together, and afterwards in year 3 we got really good results and we published in really good papers as a result of this project. I'm going to illustrate one example. So here in metabolome, you can see that we have a very complex metabolome matrix. We can have samples with different phenotypes, and each sample can have many different kind of metabolites, and they have different levels of metabolites. And then we can do the data mining by multivariate data analysis. But we can also do like a PLS model in which metabolome data can act as an explanatory variate to explain a response variate. So here, the paper for biotechnology for biofuels here by a PhD student, former PhD student who already graduated, Theo Xiaoting from my lab. This is the use of metabolomics-based strategy to identify gene targets and the application to improve 1-butanol tolerance. So of course, we know that this strain improvement is a very important goal in bioindustry, and there are many different complex phenotypes that affect the production that we need to improve. So many different strategies have been employed to achieve strain improvement. One of them is this one. Rational engineering is what has been used for many years by metabolic engineers. Based on rational engineering, we understand the system, and then we construct a rational design, and we know well about the mechanism, and we then construct the gene target and then result in improved strain. But this process takes a long time, and it's not always very useful when you don't know a lot about the microbe that you're using. And another approach is the random approach that has been used successfully in industrial microbiology for many years. We just do adaptive process or mutagenesis, for example, like laboratory evolution or transcription factor mutagenesis. And then we result in random changes, so many mutations in the genome, but then we get useful improved strain. But then it's quite difficult to get a feedback for further improvement. So the middle ground approach is the semi-rational approach using omics approach, such as comparative genomics or transcriptomics. We see the differences in genes and gene transcripts and then get some gene targets. And in this case, complete elucidation is not always necessary, but we can then get useful gene targets and understand the basic reason why these gene targets may be useful for the strain improvement. So this semi-rational strain improvement usually analyze the comparative do the comparative analysis of different levels in the system biology. And based on the omic level comparison between strains, we can get hints as to which genes, proteins may be important. And sometimes comprehensive elucidation is not necessary. So up until now, comparative studies have mostly focused on the genome or transcriptome for bioindustrial strain improvement. And, sorry, this cut here, but so sometimes it may not correlate directly to the final outcome. And the metabolome, as we know, is situated downstream in the flow of genetic information and may have more direct association with the phenotype. So we have already demonstrated that this kind of PLS model-based approach is very powerful to predict yeast replicative lifespan and also zebrafish developmental stage. And in this case, we would like to propose the usefulness of metabolomics, non-targeted metabolomics for improving 1-butanol tolerance. So this is how we do it. We cultivate the strains, and then we do the growth measurement under stress, and then we perform the metabolome analysis, and then these two data set is combined. We use metabolome data as the explanatory variate, and the 1-butanol tolerance as the response variate. And then we get the PLS model, regression model, and afterwards we select the metabolites that are strongly correlated with the phenotype, and then map that into the metabolic pathway, and then we do the genetic modification based on the corresponding metabolic pathways. So how we do it is we aim to do high throughput, many strains that have a range of phenotypes in terms of 1-butanol tolerance. So in order to do that, we construct over 100 transcription factor mutants. As we know, one transcription factor mutant deletion might affect or might give a global perturbation to the metabolism, and therefore we expect that to result in many different phenotypes. So we have 106 transcription factor knockout mutants, and then we screen them, and we select those that have non-straight growth rates close to the wild-type strain, but it has a different growth rates under stress, and this different range of 1-butanol tolerance will be very useful to make a regression model. And also, we choose the specifically metabolism-related mutants because we hope that the metabolome data as a explanatory variate can explain the differences of the phenotype. So 20 strains with varying growth rates under stress were selected for metabolome analysis and regression modeling. And then we do the cultivation and sampling in this way. We do a fast filtration for the quenching, and then the extraction was performed using methanol, chloroform, and water. And we do the GCMS analysis in scan mode. And the data processing workflow is as follows. We do the peak detection and alignment using MetAlign. And a former PhD student, Sugawa-san, from our laboratory, who developed the AI output. This is for the peak annotation. And then we do the multivariate analysis using SIMCA-P. So after manual inspection of the chromatogram, we annotated the following metabolites coming from amino acids, nucleotides, TCA cycle, intermediates, and other metabolites. So the regression models contain 76 samples and 42 variables that were used for the OPLS model. And the VIP score, this indicates that these are the variables that are important to the projection. So we selected metabolites with high VIP, and here we chose threonine and glycine, and these two metabolites have a positive PLS coefficient, which means that threonine and glycine were selected as putative important metabolites associated with 1-butanol tolerance. So we think that these two metabolites are very close in the pathway, and they have a positive correlation with the increase of 1-butanol tolerance. So in order to prove the hypothesis, if we think that threonine and glycine is important to increase 1-butanol tolerance, so we construct the several strains to increase the threonine and glycine metabolite pool inside the cell. So to increase threonine and glycine, we deleted some genes to accumulate threonine and glycine. These are the competing genes to convert threonine to, for example, alpha-ketobutyric acid, or glycine to serine. And as a result, all of the mutant strains showed increased tolerance compared to the wild-type strains, and the result is statistically significant. So in order to prove whether these mutants are well fitted back into the model, we use that newly constructed mutants, and then we want to check how well predicted values match observation for the training set. And also we check also the RMS-EP, how well predicted values match observations for the test set. So we use the newly constructed mutants as a test set. And the relationship between metabolome and the 1-butanol tolerance also holds for the new strains, which means that the increase in 1-butanol tolerance were due to the targets suggested by the model. So in summary, we can construct a regression model for tolerance using metabolome data, and the model was used to indicate important metabolites correlated with tolerance, and new tolerance strains were obtained based on the semi-rational analysis. So for the future prospect, this kind of study can be expanded upon, and we have expanded it. This one was published 2 years ago, and in 2015 and 2016, we did a similar experiment. We can improve 1-butanol productivity, and as well as titer, and also ethanol tolerance using similar strategy. And we also use different variety of analytical platforms. We construct a model using LSCMS data. In this case, we use GCMS data and obtain targets, different metabolite targets. And by using different alternative means of data analysis, we might be able to get new targets for strain improvement. So I think that is all for my talk. Probably next, Kathy will give a talk for the next session. Thank you very much.
Okay, so good afternoon, everyone. So my name is Kati. I come from Kuopio, Finland. And first of all, I would like to thank Tim and Julia for inviting here. I'm very happy to visit Australia for the first time. And we arrived already a couple of days ago. And yesterday we visited the North Stradbroke Island. And there we were able to see live kangaroos and even see the whales migrating to the north. So if you have any extra days after the conference, I can warmly recommend that place for a visit. So my topic will be mostly on the quality control approaches for metabolomics metabolomics analysis, and also very slight introduction to the quantitative aspect. Is it really quantitative to run a metabolomics analysis or not? And now that you have heard the two previous talks about the importance of the study design, what are the aspects that you need to take into account, as well as on choosing the correct analytical platform, I will mostly focus on once you have your samples ready and once you have decided which analytics you will go, then what about then to keep your analytical instrumentation in shape, and what are the aspects to focus on in terms of quality on that side. Then before I prepared my talk, I included slides about the quality on data analysis as well, but just yesterday I realized that there is actually this afternoon a whole session on this. Warwick Dunn will give a talk on this topic, so I will pass that quite fast. And then in the end a little bit about the quantitative aspect. So first of all, and now I have to say that I am completely biased to non-targeted metabolic profiling utilizing LCMS approaches. So all the information here is now on these analytics, but I'm sure more or less goes for NMR side as well. So when we talk about in general metabolomics approach, what's the aim there? What do we consider as a good quality experimental metabolomics analytics? We want to see as many metabolites as possible, that's one, if it is a non-targeted profiling. We want to have these metabolites in as high intensity signal as possible, because the higher the signal, the easier the peak picking procedure for the algorithm is, and the more chances there are that we will get a decent MSMS spectrum, and then we can identify the compound. The method we develop, we would like to keep it as reproducible as possible, because the next time we will encounter the same metabolite, we would likely to be able to identify that again then. And then to run as long sample sequences as possible. This is quite typical nowadays, especially if you move towards clinical and epidemiological studies, as you heard about the number of cases you need if you want to do an epidemiological study. Then we need to have our analytical setup in such a shape that you can run long sequences and still be confident with the results you will obtain. And in order to achieve this, it means basically on the wet chemistry on the left side, you need to give focus on the sample preparation, the chromatography, and also on the mass spec side, the detection. These all, once these are in shape, then the chances to get good quality data for the data analysis is then maximized. So first of all, what are the points to keep in mind in terms of sample preparation? Again, here, we would like to keep the pool of analytes as large as possible. But however, as we are all aware, there isn't any uniform extraction solvent so that we could get it all on one go, but merely we had to have the decide between whether it's more onto the polar side, the water-soluble components, or then more towards the lipid side that we will extract our metabolites. Then quenching, stopping the metabolism is essential. Once the sample is collected, then you don't want to have any conversions to occur in the metabolites, so all the enzymatic reactivity and any other metabolic conversions need to be stopped, and typically this is done with protein precipitation following a filtration of the sample for the analysis. And also the choice of extraction solvent typically should be such that you have minimal loss on the metabolites due to degradation or other conversions. So there are many aspects to take into account on this side. And challenges here clearly is that there is a huge variability. There is always a compromise to make when you select on the extraction solvent. And also one aspect is that, for example, in plasma sample, if you want to be absolutely certain that you have precipitated all the proteins from the sample before you inject it into your instrument, it typically means that the sample gets quite diluted. You need to add something like 4 times acetonitrile, for example, to your sample volume so that you will get the metabolites. Many laboratories then consider here evaporation and redissolving, but again, this adds analytical steps which can then cause further degradation on the metabolites and so on. So that's one of the challenges. And again, how to maintain as many different metabolite classes soluble in your sample as possible. Whereas in the side of chromatography, what kind of issues you need to take into account there? Obviously, your aim is to retain and separate, to keep all the metabolites that you have in your sample. You want to separate them so that you can then later on analyze them. And also, the method that you choose for the chromatographic separation should be such that you are still able to reproduce it, sample after sample. If you run 300 samples, you need to be sure that the first sample behaves in a similar manner than the last one. And furthermore, the mobile phase solvent that you are using should be such that it allows the ionization of as many analytes as possible in as high intensity as possible. And here clearly these are one of the among the largest challenges that you can encounter in the metabolomics experiment, how to maintain the LC side constant, because the instrumental drift is very typically occurring on both the in terms of the retention times, in terms of the intensity. And also one of the challenges is that the solvent, the method, the gradients for the chromatography should be such that even when you have isomeric species, you should be able to separate them from each other. So as few overlapping components as possible is typically desired in this side. And in about the detection then on the mass spec side, aims are clearly, as is said already many times, to maximize the number of the features that you can see, and also so that these features would be ionized in such a manner that you can rely that from sample to sample the ionization is occurring in similar manner and in such a high intensity that the identification also would be possible. And on the mass spec side then, the challenges mostly are due to, for example, ion suppression. This relates to the concentrated samples where there are thousands of analytes can be in the same sample, and these can then affect each other's ion ionization capabilities. And also the dynamic range is huge, so this is a big challenge for the instrumental side. But luckily, nowadays the latest developments on mass spec side, they are very good in coping with this. So most of the time we can have several orders of magnitude of difference in the concentration that we can encounter in one analytical run. And also, again, the isomeric species need to be able to be identified. And last but not least is the discovery of novel metabolites. This is, of course, both the sugar and salt of the metabolomics experiment, because you want to know as much as you can, but then what to do when you encounter a huge amount of such things that you cannot identify. So that's clearly one of the challenges on this side. So all of these aspects, we could spend 1 hour on each of these, how to develop these and how to optimize these. But within this lesson, we don't have time for that. So I will leave this open and clearly only want to emphasize that these are the aspects that need to be taken into account. And then just show a couple of very simple tricks, which not many of us necessarily take into account, that these also affect the outcome of the experiment. So, for example, a simple thing as injection volume. If you inject 1 microliter, you will get less features than if you inject 4 microliters. So that's simple as that. But there is also a downside. You have to remember that the more you push there, the more demanding will be the data analysis, the more demanding it will be for the algorithm to collect the peaks and to process all the hundreds of samples in a similar manner. So there is a plus and minus side in this. And the same goes also for the dilution. So if you come from the targeted route, then if you don't detect many compounds, you might think that let's dilute the sample a little bit, because then the analyte will be more clear. But this is not actually true in a metabolomics experiment. The more concentrated the sample is, the more features you will gain. And that goes for all approaches. And another thing that you can play with the mass spec side is then how the actual acquisition speed that you set up the instrument is affecting the data quality. So, for example, the faster you scan, the longer time you take to collect signals from one spectrum, the more information you will gain, but the less spectra you will collect over the time. And the more time you spend on collecting, the higher the intensity will be, for example, in the total iogram look of your data. And again, this will clearly affect the amount of components that you can collect. But similarly here, there is the downside. The more you want to get, the more variance there is in the data quality, and more emphasis needs to be on the data analytical side then. But how to control all these features during a long sample sequence run? But perhaps the best way and most often utilized is to use the pooled quality control samples, where a small aliquot from all the analytical samples is taken, a couple of microliters, 10 microliters, and they are pooled and mixed in one tube. And in this manner, your quality control sample, it is really representing your analytical samples, and it is much more reliable than if you inject some, for example, external standard mixture of 10 or 20 compounds. It can behave in a very different manner than quality control sample, which is based on completely the same matrix as your samples. This is an example from our how we typically do. We have the quality control sample. We prime the sequence with quality control samples, 10 or 20 injections of the quality control sample, so that the column and the instrumentation will be calibrated for this. You get much more stable retention time for the analysis. And then the analytical samples are randomized, typically 12 or 10 samples per set. And with this kind of sequences, in our LCMS setup, typical maximum length is around 300 injections. And after this, the signal starts, or the instrument starts to get dirty and requires maintenance. And this is more or less at the moment the maximum with our instrument that we can run on one go. And then between all the analytical samples, we have the QC samples, which are intervened throughout, and these samples we open constantly throughout the run, so that as soon as we notice that something is happening in the chromatogram, then we know that there should be some attention needs to be done. But we have avoided adding solvent blanks, because once you put in the middle of the run pure solvent there, it shakes the stability of the column easily, and these can be seen as a very slight shifts then in the QC samples. And then also the QC samples are very good for running the auto MSMS runs inside the same analytical run. And typically then at the end you can have, if you have a very different kind of samples, for example apples and bananas, you are expecting to have really different kind of metabolites present in your samples, then it's better to run the auto MSMS on also different sample types, as well as if you have any relevant standard compounds that you want to include in your analytical run. And after or during this kind of run, then this is the optimum case. Unfortunately, it is not as beautiful as this always. Probably many of you have also seen different kind of cases. But in this case, this is 236 samples, and QC samples after every 12 analytical samples. So 22 QCs overlaid, and when you see a figure like this, you can be confident that the instrumentation, the analytical setting has been in a good shape, and you can trust and rely that you have produced a good quality data to work with. And then on the data analytical side, the utilizability of the QC samples, it is probably the very first step that you will take in your data analysis. You collect the data, you run your very first PCA analysis on the data, and you check where do they locate, how do the QC samples behave. And again, this is one of the best examples that you can get. All your analytical groups are clustered nicely together, and because your QC samples are a pool of all your analytical samples, they should be nicely clustered in the middle of the PCA plot. But that's not the case most of the time. What is the use of QC samples in this kind of case? So once you open two QCs, there has been 12 analytical injections, and then you open the next QC, you see a figure like this. Then you immediately know that, okay, so there is a leakage, the column isn't stable, something is occurring. And this is a case where there is nothing to do. You need to stop the run and you need to fix the problem. And that's how you can, utilizing the QC sample, you can immediately detect this already during the run. You don't have to wait the whole sequence to check it. And also in this kind of cases, a very good checking point is to see the pressure profile of the QC samples. It should be perfectly overlaid. Already a small shift like this indicates that there is a problem in the chromatography. Then this is also a case what can happen very unfortunately many times, and this is such a thing that we haven't clarified completely why is it this way, but it is in some parts of the chromatogram the retention times aren't stable. They are moving. It can be that a couple of parts, some part is really stable, but then elsewhere there is a huge shift. And this is what happens. So if you follow this PCA plot and the sample numbers, so you see that there is in the order of sequence. So it starts from here and then goes up back and then a little bit more clustered so that it is getting more stable. So this can be both due to the column and the solvent system, something wrong there, or it can also be that if you have a very fresh samples that you put, there is still some conformational changing occurring in the sample chamber during the run. And if you encounter things like this, from my behalf, I wouldn't consider this a set that would go for the data analysis, but I would try to repeat with modifications on the chromatographic side. Then again, what you can observe from the QC samples. Here you see 2 examples of QC samples, 5 QC samples overlaid during a run. And in the case A, you see that there is a shift so that the signal is decreasing. And this is a very typical case when you know that the ion source is getting dirty or the capillary needs to be cleaned or changed, and, or some other things. If you see things like this, most of the time they refer to the problems within the mass spec side, not on the chromatographic side. And here the same QC samples were injected again, or the sequence was initiated again, and in the example B you can see that it is much more stable and overlaid. Once the instrument is again clean and nice and neat and ready to go, then the data can be more reliable then again. And then this is a warning example, or actually it underlines very much the importance of designing the experiment and also preparing very carefully for the experiment, because in this case there was a case that the person who ran this ran out of the solvent, realized that, oh dear, so the solvent isn't going to be enough for the whole sequence run. He went to the lab, decided to add a little bit, and this is what happened. So there is the beginning of the sequence with the original solvent, and then precisely the same solvent he added to the same bottle, and then there is a huge shift in the PCA plot. So this is also one of the things that planning for the experiment, also preparing enough solvent, is very important. But again, quality control samples are very good. Oh, actually, this contains all the samples, this batch. It was so clear difference that it's clear to see like this. Okay, but then on the quality side, on the data, so this I will run through quite fast because there will be much more on this topic later on. So the most important thing clearly is to normalize the data, control issues like I have shown here. The cases that I have shown, they are severe cases. Those are not good enough for data analysis. But typically there is some slight shift, some changes occurring, and these need to be controlled so that you normalize the data and you put everything to the same level so that you know that you are able to produce. So this is the manuscript contributed by David, so probably many of you know this is one of the most highly cited in this sense, this paper. I'm not going to detail this for the reason I already mentioned. And then there are also a whole lot of other aspects for the normalization side. And also what I would like to mention, a couple of things, or a few things, is that once you are working with such a rich mixtures that, for example, plasma sample, there is huge amount of different kind of metabolites present, and these metabolites, they can actually have a different kind of behavior during the run, and then it comes to a point that not a single one size fits all doesn't go anymore, so you cannot do the normalization based on the whole set, but you need to focus a little bit in detail. And this is a data from our lab published last year. And here, Carl Brunius, the first author, he generated an algorithm where the data is first divided to separate clusters, and then based on the behavior inside this cluster, then the data is corrected with the special requirements for that cluster. So you see here in the 4 different blocks that the upper side is always the actual result, and then the downside is the corrected one. So you see that there is a lot of variation between the different clusters. So if you would try to correct everything using the same factor, you would probably cause also a lot of error for the data. And then on the PCA side also, you can see that they are tightened up. A is without correction, B and C is then with correction. The red dots are quality control samples, and then the blue dots are reference samples which will run throughout different batches, and then D versus E is then after this kind of correction putting together the different batches. And the batch-to-batch correction is a very demanding task actually, so that's quite a big issue. Also in this case it isn't working perfectly, but you have to do it in any case if you run thousands of samples. So. And then this is also a topic of the afternoon, imputation. You need to correct for the zeros. You cannot enter any zeros without knowing for sure if it is a zero or if it is a missing value. And this is also one topic in the data analysis, the redundant ions. This hasn't been getting too much attention, but from our personal experience, this is a very serious topic because the feature collection algorithms that are utilized nowadays, what they do is that they collect the signals and they are not able to deduce whether they come from the same compound or not. The only thing that they do is that if there is a sodium adduct or if there is a dimer or some simple modification that you can computationally program for the algorithm, then it's fine. But in many cases, this is not the case. So there is a redundant representation of the same component in the data matrix. For example, this is a peak of proline. So we use Agilent software, and in this case, the Agilent software collects all the signals which are actually different modification adducts and different forms of the proline compound. And then here on the yellow you see, so this is a short screenshot from the data output from Excel. And the red line is the actual proline compound, but then when you look the data in a little bit more wider scope, you see that all these yellow lines, they also represent proline. So instead of having proline once there, you have it like 10 times. And you can imagine what this does when you take your data matrix and do the false discovery correction if you have many different metabolites represented as 10 different ions. And this is actually one quality aspect of metabolomics data that hasn't gained as much attention as it should. And for example, our group is working on this aspect at the moment. So there should be more switch from reporting features towards reporting compounds, actually. And then a very brief introduction on the quantitative aspect. So this is a very schoolbook chemistry. You all know that if you want to quantitate something, you made a calibration curve, a standard mixture. But if you want to do this on metabolomics, on targeted metabolomics side, then you need to have such a matrix where your analyte doesn't exist. For example, if you give some drug to a person, then it is possible to establish the method on urine sample because urine typically doesn't contain that drug, and then you can measure. But in majority of the cases, the compounds that you want to measure quantitatively, they already exist in your sample matrix. And in this kind of cases, you can add internal standard which can be as similar as possible to your analyte, so that you assume that it behaves in the analytical run in a similar manner as your analyte, and then based on this internal standard addition, you can then correct for any potential matrix effects that can occur. For example, you cannot be sure that if you have 5 times more of your standard compound or the analyte that you want to measure, if the signal really is 5 times more higher, or whether there is some matrix ion suppression or something in the ionization occurring that would twist or interfere with this ionization. But based on the addition of internal standards, you can correct errors like this. And then, as a last point here, is with the spiking with stable isotope labeled internal standards. This is more or less the norm nowadays. If you really want to go towards very sophisticated quantitative metabolomics analysis, you should purchase or you should synthesize stable isotope label standards so that it is precisely the same compound, the same analyte that you want to focus on, but there is typically carbon-13 or deuterium ion in that compound, so that you can differentiate those, but still they are behaving exactly the same way, and then you can use this for correction. But this is already going very much towards the purely quantitative analysis, and there is a huge amount of literature available on this side, and actually very strong rules and guidelines apply on this side if you want to have really validated method. But we are not going to this side too much. But then the question, what I am asked many times, that what about then so it's semi-quantitative. What does it mean? Is it really quantitative? Can you really trust it? What is this metabolomics? It's so too vague. People don't like it many times. But this is an example from a compound called CMPF. It's a furan fatty acid that enters our circulation if we eat a lot of fish. And we had a non-targeted metabolic profiling study utilizing QDOF instrumentation. We found this biomarker of fish intake. And here in this scheme, the orange line represents the QDOF analysis from a metabolic profiling assay. Each sample here is an individual human sample that was measured. And then the blue line represents the very same samples measured with a quantitative method that was set up on triple quadruple instrument. So purely quantitative analytical method. And as you can see, more or less the signal remains on the stable level. So there isn't too much variation. In some cases, yes, but in general, the scheme is in the same level. And if you look at the correlation, the untargeted versus the targeted method, then the correlation between these samples is good enough to say that the metabolomics, non-targeted methods as well, can give you quantitative data as well, if you don't need the exact amount, but you are happy with the relations, that how they occur. So with this, I come to the conclusion, and I hope I have shown you today that issues related to sample preparation, chromatography, and mass spec detection affect the analytical quality, and you should pay attention on developing these methods as much as you should also do it on the sample preparation and study setup side as well. And then once these are optimized, utilize the QC samples, because they give so many different aspects how you can then monitor the quality of your analytical run, and also from the data pre-processing side. And as I mentioned, from the data analytical side, we should move more towards reporting the compounds itself, not the redundant spectral features, and this still requires a lot of work to be done on the pre-processing of the data than what is available at the moment in any available platforms. And when doing quantitative analysis, or if it is enough that you have the relative quantities, if you want to know if these people have more, for example, the CMPF than these, if you are fine with the fold change analysis, then QDOF is quantitative. The metabolic profiling is quantitative enough. But if the absolute quantities need to be taken into account, then the best is to go for stabilized or labeled internal standards. So with this, I thank you for your attention.